My name is Oliver Deusch. I'm Scientific Product Manager at Computomics, and I'll talk today about successful metagenomics data management and analysis with our products Morpheus and MEGAN6 Ultimate Edition. So before I start, let me quickly remind you of the challenges that researchers are usually facing with metagenomics data. So this is about handling and storing terabytes of data, choosing a sequence data analysis tool that delivers reproducible results, the general user-friendliness of bioinformatics software, or rather the lack thereof. Then there's functional and biochemical pathway analysis, there's data visualization and result interpretation, and then there's statistical and machine learning analyses that need to be carried out. And then there's comparisons with historic studies, and also what's becoming more important today is collaborating with remotely located colleagues. And when you're a microbiologist and you're performing a microbiome analysis, you're very good at your result interpretation because you know your microbes and you know what changes in the microbial composition mean. But you're also faced with all those other challenges from fields such as bioinformatics, statistics, information technology, and computer science. And you may not be an expert in those areas. And the goal with our solutions really is to make microbiome research more accessible so that you can concentrate on the aspects of the work that you do best. So these are the three pillars of our metagenomics products and services. So we have Morpheus, which is a web-based comparison and analysis platform that can do the data processing and storage for you. And when you're using Morpheus, you don't need local compute infrastructure. Obviously, you need a web browser to interact with the platform, but I'll take that as a given. Then there is MEGAN6, which is a comprehensive microbiome analysis software tool that runs on desktop computers. And the most important feature here is that MEGAN6 has KEGG pathways built in so that you can go easily, or you can go straight into functional analysis. We also offer customized metagenomics analysis service, and this is why we really where we work closely with our customers to understand their problems or their research question, and then analyze the data for you. So I'll start introducing MEGAN. Many will be probably already familiar with this. So to my knowledge, it's the only interactive metagenomics analysis tool. And depending on your data, MEGAN offers strain level resolution. Since its release, it's been cited more than 4,000 times in the scientific literature. And a bit more recently, long read support has been added. So if you're working with PacBio or Oxford Nanopore reads, MEGAN is compatible with that. So typically, you would perform visualizations and analyses in MEGAN through a graphical user interface. But MEGAN also offers command line tools and scripting functionality. And this is really that way you can really combine the best of both worlds. So a GUI on one hand side and also scripting and automating on the other side. You can always export your data. So if you choose to perform additional analyses, you can always export data tables. And as I said, you have KEGG pathways built in so that you can go straight into functional analysis. I'm not going to go into detail about those technical features, but let me just say that MEGAN works really well with Diamond, which is a very fast protein alignment tool. So we have a very streamlined workflow, and it's really quick. You can get really quick turnaround times using Diamond integration, and you have very similar sensitivity compared to BLAST. So this is a screenshot of MEGAN, and this shows you a taxonomic tree. So this is the NCBI backbone tree, which can be collapsed and uncollapsed at different ranks of the taxonomy tree. And you can overlay the abundance of your of the respective taxa in your samples. So in this example, we have 4 samples, and you can overlay the abundance in your samples of the respective taxa onto this tree. Now, the same thing works for functional comparisons. So this is a biochemical pathway taken straight out of KEGG, and you can overlay the abundance of the respective enzymes onto this pathway map. And for example, here you can see that this enzyme 4211, with this enzyme condition number 4211, this is abundant in the blue and in the yellow sample, but it's not present in the other samples. So this can be a really good start for further analyses, and you don't need to take any extra steps. So with the same data processing that is required to do the taxonomic analysis, you can already start with the functional analysis, because it's all handled internally with MEGAN. Okay, so this is our recommended data analysis workflow for MEGAN. So you start with raw sequence data from your samples. You align them against the reference database. We recommend NCBI-NR because it's the largest database available. Using Diamond, so it's nice and quick. You generate alignment files. The alignment files need to be MEGANized, and then they can go straight into MEGAN for interactive analysis. You can provide your study metadata, and that makes the comparisons obviously more powerful. Now, although this is a relatively streamlined workflow, it may still be not feasible for some users, because either you're not very good at bioinformatics, so you're struggling to run your Diamond analysis, for example, or you may not even have a compute infrastructure to run these steps on. And this is where Morpheus comes in. So Morpheus is our web-based platform that handles data storage, processing, analysis, and visualization. And I'm not going to go into all of those features and benefits listed here. I'd rather show you a quick overview and give a quick live demo, so you get a better understanding of what the platform can do for you. So this is an overview of how researchers can interact with Morpheus. So on the right side is the client, that's you. You start with your samples, you ship them to a sequencing lab. They perform DNA extraction, library prep, and sequencing. And then the sequence data is uploaded onto the Morpheus servers. So, and what happens here is that our automated data analysis pipeline kicks in. Our pipelines are pre-computed. Data storage also happens here, and you can leave your data on here if you choose so. But if once the analysis is finished, you get a notification. You log into this platform, and then you can immediately start looking at interactive reports. And this is then really the first time you interact with when you get your data back, the first time you interact with it, you're already looking at results, and you're not handling large sequence files. You can provide study metadata, so you can perform some more meaningful comparisons here. You can always export your data tables for further analysis, but you can also download a MEGAN-compatible comparison file so that you can easily go and do further analyses in MEGAN. So let me give a quick live demo of the platform. Let's hope this works. I'll go to my web browser, and yeah, so once you receive your notification that your data has been processed, you go to our login page, you log in, you are presented with this main page, which features all the applications that we have to interact with your data. So for example, you click at the most abundant taxa visualization. You choose a phylogenetic or a taxonomic rank, I should say, of your liking, for example, the genus level. And then in combination with the metadata that you're providing, you can sort your plot. So here we have 2 diet groups. And if you sort by diet, you have 1 diet group on the left and 1 diet group on the right, and you immediately see differences. For example, this orange taxon here is only ever present in the right diet group and not on the left side. And what this really means is that you've just logged onto the system, and this is the first time you're interacting with your data. Once you get your results back, obviously you have to have provided metadata, but this is the first time you get, after getting your results back, and you can already draw conclusions, and you're not still wrestling with large sequence files. So in the interest of time, I'm just going to go briefly through the other apps that you have on here. So we have a PCA functionality, again, where you can color your data by metadata. Various diversity analyses are possible here with different diversity indices. Again, you can use your metadata to group your samples. And then I'm just going to show you one more. So you've got a very sophisticated data upload and download functionality, not just for single files through your web browser, but also for large data sets through command line interfaces. And also, and the last plot I'd really like to show you very briefly is actually not that one, but correlations of taxa and functions. And here you can choose taxonomic rank again, and you can choose a pathway, and you get a visualization of path or taxa that correlate with certain pathways. Okay, so I'm going to go back to my slides and finish this brief presentation. So I just gave a very quick overview of our metagenomics products and services. So Morpheus, the web-based platform, MEGAN, the tool, the standalone tool for metagenomics analysis. And I didn't even explain our detailed analysis as a service. So hopefully this was of interest. If you have any questions about those products, reach out to me or one of my colleagues during this meeting. And yeah, thanks for your attention, and I'm taking any questions.